The phenomenon of nocturnality represents one of the most profound adaptations in the history of life on Earth, a fundamental restructuring of physiology, behavior, and sensory systems that has occurred independently across virtually every major animal lineage and continues to shape ecological communities on every continent except Antarctica. Understanding why some animals have evolved to conduct their lives primarily during the hours of darkness requires moving beyond simple characterizations of night dwellers as creatures of shadow and mystery toward a rigorous examination of the evolutionary pressures, anatomical modifications, physiological constraints, and ecological relationships that together explain this remarkable pattern of temporal niche partitioning. The story of nocturnal animals is ultimately a story about competition, predation, thermoregulation, and the exploitation of resources that remain unavailable or inaccessible to organisms operating under different temporal regimes. And tracing the development of scientific understanding of nocturnality reveals how researchers across disciplines from comparative anatomy to molecular biology have gradually assembled a comprehensive picture of life after dark. The very definition of nocturnality involves more complexity than might initially be apparent, as animals exhibit a continuous spectrum of activity patterns rather than falling neatly into discrete categories. Strictly nocturnal species confine their activity almost exclusively to the period between sunset and sunrise, while crepuscular animals concentrate their movements during the twilight hours of dawn and dusk, when light levels remain intermediate between full daylight and complete darkness. Cathemeral animals distribute their activity throughout the 24-hour cycle, without showing consistent preference for any particular phase, and many species display flexibility in their temporal patterns depending on season, reproductive status, food availability, or the presence of predators and competitors. The biological mechanisms underlying these patterns involve the circadian system, an internal timekeeping apparatus present in virtually all organisms that synchronizes physiological and behavioral processes with the external light-dark cycle. And the study of these mechanisms has revealed that the distinction between diurnal and nocturnal animals extends far deeper than simple differences in when animals happen to be awake. The circadian clock operates at the cellular level through feedback loops involving clock genes whose protein products regulate their own transcription over approximately 24-hour cycles. And while this basic molecular machinery shows remarkable conservation across species from fungi to humans, the way these internal clocks interact with external time cues differs systematically between animals adapted to different temporal niches. The suprachiasmatic nucleus in mammals serves as the master pacemaker that coordinates rhythms throughout the body, receiving direct input from specialized photoreceptive cells in the retina that detect environmental light and adjust the phase of the internal clock accordingly. But the downstream signals that this central oscillator sends to regulate behavior and physiology are essentially inverted between nocturnal and diurnal species. Research into the neural pathways connecting the circadian system to sleep-wake regulation has demonstrated that nocturnal animals are not simply diurnal animals with reversed clocks, but rather possess fundamentally different wiring in the brain regions that translate circadian signals into behavioral output, a finding that has significant implications for understanding the evolutionary transitions between diurnal and nocturnal lifestyles. The evolutionary origins of nocturnality extend deep into the history of life, and reconstructing the ancestral activity patterns of major animal groups requires integrating evidence from multiple sources, including the fossil record, comparative anatomy, molecular phylogenetics, and ecological modelling. The nocturnal bottleneck hypothesis proposes that the common ancestor of all living mammals was likely nocturnal, driven to adopt nighttime activity by the ecological dominance of dinosaurs during the Mesozoic era. And this hypothesis draws support from the observation that many apparently conserved mammalian traits appear to represent adaptations to life in low-light conditions. The relatively small body size of early mammals, their well-developed auditory systems, the initial loss of colour vision capabilities that were only later partially recovered in some lineages, and the sophisticated olfactory apparatus characteristic of the group all make sense as features that would prove advantageous to animals operating primarily at night. Fossil evidence regarding activity patterns remains inherently limited, since behaviours do not preserve directly, but researchers have developed methods for inferring probable activity patterns from features of fossil skulls, including the size of the orbit relative to skull length, the dimensions of the optic foramen through which the optic nerve passes, and the morphology of the sclerotic ring in species where this bony structure surrounding the eye has been preserved. The end Cretaceous mass extinction that eliminated non-avian dinosaurs approximately 66 million years ago appears to have released ecological constraints on mammalian evolution, and the subsequent radiation of mammals into diverse ecological roles included transitions back to diurnal activity patterns in multiple lineages. The anthropoid primates that include monkeys, apes, and humans evolved from nocturnal ancestors but subsequently returned to daytime activity, a shift reflected in the reacquisition of trichromatic colour vision through gene duplication events that restored some of the visual capabilities that had been lost during the nocturnal bottleneck. This pattern of ancestral nocturnality, followed by secondary diurnality, appears to characterise several major mammalian lineages, while other groups, including many rodents, bats, and carnivores, retained the nocturnal habits of their ancestors. The evolutionary transitions between temporal niches have not been unidirectional, as some lineages show evidence of multiple shifts in activity patterns over their evolutionary history, suggesting that the selective pressures favouring particular temporal niches can change depending on competitive relationships, predation risk, and environmental conditions. The sensory adaptations that permit effective functioning in low-light environments represent perhaps the most extensively studied aspect of nocturnal biology, with vision receiving particular attention due to the obvious challenges that darkness poses for visual organisms. The vertebrate eye contains two fundamental classes of photoreceptor cells: rods specialized for sensitivity at low light levels, and cones adapted for color vision and high acuity in bright conditions, and the relative proportions and distributions of these cell types differ dramatically between nocturnal and diurnal species. Nocturnal animals typically possess retinas dominated by rod photoreceptors, with cone cells constituting only a small fraction of the total receptor population, a proportion that may be inverted in strongly diurnal species. The rod photoreceptors of nocturnal species often display specialized features that enhance their sensitivity, including longer outer segments containing greater amounts of the light-sensitive pigment rhodopsin and integration times that allow summation of photons over extended periods at the cost of temporal resolution. Beyond the receptor level, the neural processing of visual information shows systematic differences between nocturnal and diurnal species that reflect trade-offs between sensitivity and resolution. The convergence of signals from multiple photoreceptors onto single retinal ganglion cells increases sensitivity by pooling photons captured across a larger area of the retina, but this spatial summation necessarily reduces the ability to resolve fine spatial detail, and nocturnal animals generally show higher convergence ratios than their diurnal counterparts. The optics of the eye itself contribute to light-gathering ability, with nocturnal species typically possessing larger eyes relative to body size and larger pupils that can open wider to admit more light under dim conditions. The tapetum lucidum, a reflective layer behind the retina found in many nocturnal mammals, increases visual sensitivity by reflecting light that has passed through the retinal photoreceptors back for a second chance at absorption, and this structure produces the eye shine visible when nocturnal animals are illuminated at night. The tapetum appears to have evolved independently in multiple lineages, with different groups employing different reflective materials, including guanine crystals in many fish and collagen fibers in certain ungulates, demonstrating the convergent evolution of solutions to the common problem of operating in light-limited environments. The extreme case of visual specialization for nocturnal life appears in the tarsiers, small primates found in Southeast Asian islands, whose enormous eyes constitute the largest eyes relative to body size of any mammal, and indeed approach the physical limits of what is possible given the constraints of skull anatomy. Each eye of a tarsier has a diameter greater than that of its brain, and these organs provide the remarkable light-gathering ability necessary for a visually oriented predator operating in the forest understory at night. The owl family presents analogous adaptations in birds, with forward-facing eyes of exceptional size that provide the binocular vision useful for judging distances to prey while maximizing sensitivity through expanded pupil area and densely packed retinal photoreceptors. The tubular shape of owl eyes unlike the spherical eyes of most birds, represents a solution to the challenge of maximizing corneal diameter within the space available in the skull, though this specialization comes at the cost of eye mobility, such that owls must turn their heads rather than their eyes to look in different directions, a behavior facilitated by the remarkable rotational ability of the owl neck with its increased number of cervical vertebrae. Not all nocturnal animals rely primarily on vision, and the elaboration of non-visual sensory systems represents an equally important category of adaptation to darkness. The extraordinary hearing abilities of many nocturnal species have attracted scientific attention since the 19th century, with owls again providing some of the most remarkable examples of auditory specialization. Barn owls can capture prey in conditions of complete darkness using sound localization alone, a capability that depends on asymmetric placement of the ears on the skull that creates slight differences in the timing and intensity of sounds arriving at each ear, differences that the nervous system uses to compute the spatial location of sound sources with exceptional precision. The facial disc of feathers characteristic of owls functions as a parabolic reflector that channels sounds toward the ear openings, and behavioral experiments using captive birds have demonstrated that barn owls can strike prey with angular accuracy of approximately 2 degrees in complete darkness, a level of precision that requires extraordinary neural machinery for processing temporal differences measured in microseconds. Echolocation represents perhaps the most sophisticated non-visual sensory system found among nocturnal animals, having evolved independently in bats and in several lineages of cave-dwelling birds including oil birds and some swiftlets. The biosonar of bats involves the emission of ultrasonic calls, typically through the mouth or nostrils, and the analysis of returning echoes to construct a detailed perceptual representation of the surrounding environment. The complexity of the system becomes apparent when considering the computational challenges involved. A flying bat must emit calls at rates sometimes exceeding 100 per second during the terminal phase of prey capture, while simultaneously analysing echoes that return with delays measured in milliseconds and intensity diminished by the inverse square law, extracting information about target distance, direction, size, texture, and movement from these acoustic returns, while itself moving through three-dimensional space. Different bat families have evolved remarkably different echolocation strategies, with some species producing brief broadband clicks containing a range of frequencies that provide detailed spectral information about target structure, while others emit longer constant frequency calls that allow precise measurement of Doppler shifts caused by the relative motion of bat and target. The evolution of echolocation required not only the development of specialized vocalizations, but also the modification of the auditory system to process ultrasonic frequencies and the neural machinery to coordinate call emission with echo-analysis while suppressing the response to the outgoing call itself, which would otherwise overwhelm the returning echo. The middle ear of echolocating bats includes specialized muscles that contract briefly to dampen sensitivity to the emitted call while allowing rapid recovery in time to detect the echo, a process requiring precise timing coordination with the laryngeal muscles that produce the call. The auditory cortex of echolocating bats contains systematic representations of acoustic parameters relevant to target localization and identification, with different cortical areas specialized for processing information about target distance, velocity, and fine structure, demonstrating a level of neural specialization comparable to that found in the visual cortex of highly visual species. Olfaction provides another crucial sensory channel for nocturnal animals, and the persistence of odor trails through space and time makes chemical information particularly valuable when visual contact with conspecifics, prey, or predators becomes impossible. The olfactory systems of many nocturnal mammals show expansion relative to diurnal relatives, both in the peripheral apparatus of olfactory receptor neurons, in the nasal epithelium, and in the brain structures that process olfactory information. The main olfactory epithelium contains neurons expressing a diverse array of odorant receptor proteins, each neuron typically expressing just one receptor type from a repertoire that can number over 1,000 in species with well-developed olfactory capabilities, and the signals from neurons expressing the same receptor converge on specific glomeruli in the olfactory bulb, where initial processing of odor information occurs. A separate vomeronasal system detects pheromones and other social odours through a distinct set of receptors and neural pathways that project to brain regions involved in reproductive and aggressive behaviours. And this accessory olfactory system shows particular elaboration in nocturnal species that rely heavily on chemical communication. The star-nosed mole presents perhaps the most remarkable example of non-visual sensory specialisation, possessing a ring of 22 fleshy appendages surrounding the nostrils that contain more than 25,000 sensory receptors called Emer's organs, making the star the most sensitive known touch organ in any mammal. This bizarre structure allows the mole to identify and consume prey items in complete darkness faster than any other known mammalian predator, with the entire sequence from contact to consumption sometimes requiring less than a quarter of a second. The neural representation of the star in the somatosensory cortex of the mole occupies a disproportionately large area of the cortical surface, analogous to the expanded representations of the hands and lips in the human cortical homunculus, demonstrating how sensory specialization is reflected in neural organization. Whiskers, or vibrissae, provide another tactile channel of particular importance to nocturnal mammals, with elongated facial whiskers found in groups from rodents to carnivores to seals serving as active sensing organs that detect mechanical forces during contact with surfaces and objects. The follicle of each whisker contains multiple types of mechanoreceptors that respond to whisker deflection with exquisite sensitivity, and the somatosensory cortex of species with well-developed whisker systems contains barrel fields where each barrel represents a single whisker in a topographic map that preserves the spatial arrangement of the whisker array. Rats and mice actively move their whiskers in rhythmic sweeping motions called whisking that generate detailed tactile information about nearby objects, and behavioural studies have demonstrated that these animals can discriminate between surfaces differing in texture by as little as 30 micrometers using whisker touch alone. The physiological adaptations of nocturnal animals extend beyond sensory systems to encompass virtually every organ system, with thermoregulation presenting particular challenges and opportunities for animals active during the cooler nighttime hours. Small nocturnal mammals face especially demanding thermal constraints because their high surface area to volume ratios result in rapid heat loss, and many species have evolved specialized metabolic strategies to cope with these demands. Torpor, a controlled reduction of body temperature and metabolic rate, allows many small nocturnal mammals to conserve energy during periods of cold or food scarcity, with some species entering brief bouts of torpor on a daily basis during the daytime rest period, while others display extended hibernation lasting weeks or months. The physiological mechanisms underlying torpor involve coordinated changes in cardiovascular function, ventilation, and cellular metabolism, with heart rates dropping from several hundred beats per minute to fewer than 10, and body temperatures falling to within a few degrees of ambient in deeply torpid animals. Desert-dwelling nocturnal species face a different set of thermal challenges, with extreme heat during daylight hours making daytime activity potentially lethal, while cooler nighttime temperatures permit activity without overheating. The kangaroo rats of North American deserts exemplify the suite of adaptations that permit survival in arid environments, combining nocturnal activity patterns with specialized kidney function that allow survival without ever drinking water, metabolic pathways that produce water internally as a byproduct of carbohydrate and fat metabolism, and behaviors including underground burrow construction and seed caching that minimize water loss and provide access to more humid microenvironments. The shift to nocturnal activity in desert environments represents an ecological strategy for avoiding thermal stress that has evolved convergently in desert communities worldwide, with the result that nocturnal diversity often exceeds diurnal diversity in the most extreme desert environments. The behavioral ecology of nocturnal animals encompasses a vast range of feeding strategies, social systems, and reproductive patterns that in many cases remain less thoroughly documented than those of diurnal species due to the obvious challenges of conducting behavioral observations in darkness. The development of night vision technology, acoustic monitoring, automated camera traps, and increasingly sophisticated biologging devices that record the activities of free-ranging animals has progressively expanded knowledge of nocturnal behavior revealing patterns that sometimes challenge assumptions derived primarily from diurnal models. The hunting strategies of nocturnal predators often emphasize ambush and stealth over pursuit, with many nocturnal carnivores possessing relatively shorter limbs and more robust builds than diurnal pursuit predators, though exceptions exist among species like the African wild dog that sometimes hunt at night, and the Australian dingo that shows flexible activity patterns. The relationship between prey and predator activity patterns generates complex evolutionary dynamics, as prey species evolve to avoid periods of maximum vulnerability, and predators respond by shifting their own activity schedules. This temporal arms race has produced some remarkable patterns, including the adjustment of activity timing by prey species in response to the presence or absence of predators, as documented in detailed studies of vole populations exposed to different predator regimes. The fear of predation can influence prey behaviour as profoundly as actual predation pressure, with prey species often avoiding optimal foraging sites during periods when predator activity peaks, even when direct encounters remain relatively rare. The indirect effects of these predator-induced behavioural changes can propagate through ecosystems in ways that influence vegetation structure, nutrient cycling, and the abundances of species that might appear only distantly connected to the original predator-prey relationship. Social organisation among nocturnal animals spans the full range from strictly solitary species that interact with conspecifics only for mating to highly social species that live in complex groups with elaborate communication systems. The challenges of maintaining social bonds in darkness have driven the evolution of communication channels that function effectively without visual contact, and nocturnal social species typically rely heavily on vocalizations, scent marking, and tactile interactions to coordinate group activities and maintain social relationships. The primates offer instructive comparisons, with the largely solitary nocturnal prosimians contrasting with the more social diurnal monkeys and apes in patterns that some researchers have linked to the sensory constraints of their respective activity periods. Though the relationship between activity pattern and social system involves multiple factors, and numerous exceptions complicate simple generalizations. The owl monkeys of Central and South America represent the only truly nocturnal anthropoid primates, having secondarily evolved nocturnality from diurnal ancestors, and their social system of pair-bonded monogamy with extensive male parental care differs markedly from the more variable social systems of related diurnal monkeys. Whether this social system represents an adaptation to the nocturnal niche or reflects phylogenetic constraints inherited from ancestral conditions remains a matter of ongoing investigation, but the owl monkeys provide a valuable natural experiment in the consequences of the evolutionary transition to nocturnality within a lineage of otherwise diurnal primates. Comparative studies across the primates have revealed that the few nocturnal species in the group tend to have relatively smaller social groups, less elaborate visual displays, and more developed olfactory and acoustic communication than their diurnal relatives, suggesting consistent effects of activity pattern on social evolution despite the limited number of independent origins of nocturnality in the order. The reproductive biology of nocturnal animals includes numerous specializations related to the temporal organization of their lives, with mating behaviors often concentrated during particular phases of the night, and coordination between potential mates accomplished through signals that function in darkness. The elaborate vocal displays of many nocturnal species serve to advertise the location and quality of potential mates, with the songs and calls of nightjars, nightingales, and many other nocturnal birds providing examples of acoustic advertisement that can transmit across considerable distances without requiring visual contact. The complex ultrasonic songs of some bat species, produced in frequencies too high for human hearing, appear to function in mate attraction and territorial defense in ways analogous to birdsong, and recent technological advances allowing detailed recording and analysis of these vocalizations have revealed previously unsuspected complexity in mate acoustic communication. Bioluminescence, the production of light through chemical reactions within living organisms, occurs in numerous nocturnal invertebrates, where it serves functions including mate attraction, prey luring, and predator defense. The fireflies present the most familiar example, with species-specific patterns of light flashes serving to attract mates in courtship displays that have been thoroughly documented through extensive field studies spanning more than a century. The luminescent organs of fireflies contain specialized cells called photocytes that produce light through the oxidation of luciferin, a reaction catalyzed by the enzyme luciferase in the presence of ATP and oxygen. And the control of flash patterns involves both neural signals and modulation of oxygen delivery to the light-producing cells. Different firefly species employ distinct flash patterns that minimize cross-attraction between species, with females typically responding only to flashes matching the temporal pattern characteristic of conspecific males. Though females of some Photurus fireflies have evolved the ability to mimic the flash patterns of other species to lure males of those species as prey in a remarkable example of aggressive mimicry. The ecological roles of nocturnal animals extend far beyond their interactions with other nocturnal species to include crucial connections with diurnal organisms that share the same habitats and communities. Pollination by nocturnal visitors, including bats, moths, and certain beetles, represents an essential ecosystem service for many plants that have evolved flowers specialized for night-active pollinators, typically displaying pale coloration visible in low light, strong fragrances that function as long-distance attractants, and nectar production timed to coincide with the activity periods of their pollinators. The columnar cacti of American deserts provide a well-studied example of dependence on nocturnal pollinators, with multiple species relying primarily or exclusively on nectar-feeding bats for pollination and producing flowers that open at night, emit strong odours attractive to bats, and close by morning before diurnal visitors become active. The mutualistic relationships between night-blooming plants and their nocturnal pollinators appear to have influenced the evolutionary diversification of both partners, with some lineages of plants showing extensive radiation in association with the availability of particular pollinator groups, and some pollinator lineages showing corresponding radiation in association with the exploitation of particular plant resources. The hawk moths, with their elongated tongues that can reach nectar at the bottom of deep tubular flowers, have engaged in coevolutionary relationships with numerous plant species that have evolved correspondingly deep flowers, accessible only to pollinators with sufficiently long tongues, a pattern famously predicted by Darwin in the case of the Madagascan orchid and Gracum sesquipedale, before the moth pollinator was actually discovered. Seed dispersal by nocturnal frugivores including fruit bats and certain rodents contributes to forest regeneration and the spatial distribution of plant populations in ways that often complement the dispersal services provided by diurnal frugivores. Fruit bats transport seeds across distances that can exceed tens of kilometers, and their tendency to defecate while flying or while roosting in locations distant from the parent tree promotes the colonization of sites far from existing populations. The gut passage through bats may also influence germination success, with some studies suggesting that scarification of seed coats during digestion enhances germination rates for certain species. The loss of nocturnal seed dispersers from tropical ecosystems due to hunting and habitat destruction has raised concerns about potential consequences for forest composition and dynamics, with some models predicting significant shifts in tree species abundances following the removal of key dispersers. The predatory activities of nocturnal animals exert strong top-down effects on populations of prey species and, through trophic cascades, on the communities and ecosystems in which those prey species are embedded. Owls consume enormous numbers of small mammals, including many rodent species considered agricultural pests, and studies calculating the economic value of pest control services provided by owl populations have generated estimates reaching millions of dollars annually for intensively farmed regions. The installation of owl nest boxes to augment breeding populations in agricultural landscapes represents an increasingly common practice aimed at harnessing these predatory services, though the effectiveness of such programs depends on factors including prey densities, habitat quality, and the availability of alternative nest sites in the surrounding landscape. The predation by bats on nocturnal insects provides another ecosystem service of substantial economic importance, with insectivorous bat populations in agricultural regions consuming quantities of crop pest insects that would otherwise require chemical control. A widely cited estimate suggested that the pest control services provided by insectivorous bats to American agriculture exceed $3 billion annually, a figure that, while subject to considerable uncertainty and methodological critique, nonetheless indicates the scale of benefit that nocturnal predators provide to agricultural systems. The potential disruption of these services by factors affecting bat populations, including habitat destruction, disturbance of roost sites, and the spread of white-nose syndrome in North American bat populations, has motivated conservation efforts aimed at protecting bat populations and the services they provide. The decline of bat populations in eastern North America following the arrival of white-nose syndrome, a fungal disease caused by Pseudogymnascus destructans, represents one of the most severe wildlife disease events in recorded history, with mortality rates exceeding 90% in some hibernating colonies, and cumulative population declines reaching into the millions of individuals. The disease organism appears to have been inadvertently introduced from Europe, where bat populations harbour the fungus without experiencing comparable mortality, and the mechanisms underlying the difference in disease outcomes between European and North American bats remain subjects of active investigation. The catastrophic mortality has precipitated declines across multiple species of hibernating bats, and has created conservation challenges that extend beyond the immediate disease impacts to include potential effects on the ecological functions that these bat populations perform. The conservation of nocturnal animals faces distinct challenges related to the difficulty of monitoring population trends in species that are active when researchers typically are not, and the development of effective survey methods for nocturnal species has required innovation in acoustic monitoring, camera trapping, and other technologies that allow data collection without direct observation. Bat populations are increasingly surveyed using acoustic detectors that record ultrasonic echolocation calls, allowing species identification and activity quantification across multiple sites simultaneously without requiring human presence during the survey period. The analysis of these acoustic recordings has benefited from advances in machine learning that have automated aspects of species identification that previously required laborious manual classification, though the reliability of automated identification varies across species and call types, and human verification remains important for many applications. Light pollution represents an increasingly recognized threat to nocturnal animals, with artificial illumination at night fragmenting habitat, disrupting orientation and navigation, altering predator-prey relationships, and interfering with reproductive behaviors that depend on natural darkness. The global extent of artificial night sky brightness has been mapped using satellite observations, revealing that a substantial fraction of the world's land surface experiences levels of night illumination that may affect nocturnal organisms, with the most severe light pollution concentrated in urban areas and surrounding suburbs, but with significant sky glow extending into regions far from major population centres. The effects of artificial light on nocturnal animals vary across species and depend on factors including the intensity, spectral composition, and spatial distribution of light sources, with some species showing attraction to lights and others showing avoidance, and with some effects appearing at light levels far below those found in urban environments. Insects attracted to artificial lights may suffer direct mortality through collision with light fixtures, entrapment, or exhaustion, and the aggregation of insects around lights may draw predators to these locations while depleting insect populations in surrounding unlit areas. The navigation systems of nocturnal migratory birds can be disrupted by artificial light, with birds attracted to illuminated structures sometimes colliding with buildings or becoming disoriented and circling until exhausted. The orientation of sea turtle hatchlings, which normally navigate from beach nests to the ocean by moving toward the brightest horizon, can be disrupted by artificial lighting that causes hatchlings to move inland toward light sources rather than seaward, often with fatal consequences. The spectral composition of artificial lighting influences its effects on nocturnal wildlife, with shorter wavelength light including the blue-rich output of many LED fixtures generally producing stronger effects on circadian systems and orientation behaviours than longer wavelength light, a finding that has motivated recommendations for the use of amber or red-shifted lighting in areas where wildlife impacts are of concern. The study of nocturnal animals has been transformed by technological developments that have progressively expanded the ability to observe, track, and monitor species that operate in conditions that exclude direct human observation. Infrared imaging allows visualization of thermal contrasts in complete darkness, revealing the activities of warm-blooded nocturnal animals against cooler backgrounds without the disturbance that visible light might cause. Radiotelemetry, GPS tracking, and satellite-based location systems have enabled researchers to follow the movements of nocturnal animals across landscapes and through seasons, revealing patterns of habitat use, migration, and ranging behavior that would remain invisible to conventional observation methods. Accelerometers and other motion-sensing devices attached to animals provide detailed records of activity patterns and behavioral states that can be analyzed to reconstruct how individuals allocate time across different activities, allowing investigation of questions about energy expenditure, behavioral strategies, and responses to environmental variation. Acoustic monitoring has proven particularly valuable for nocturnal species that vocalize frequently, with automated recording devices deployed across landscapes, capturing the sounds of nocturnal birds, mammals, amphibians, and insects over extended periods and generating data sets that can be analyzed to track species presence, activity levels, and in some cases individual identity. The application of bioacoustic methods to study the complex vocal behavior of nocturnal species has revealed patterns of individual variation, learning, and cultural transmission that were previously suspected but difficult to document, and the integration of acoustic data with other information about habitat conditions, weather, and the activities of other species has provided insights into the ecological context of vocal behavior. The molecular tools of modern genetics and genomics have opened new avenues for investigating nocturnal animals, from the analysis of circadian clock genes that govern temporal organization to the identification of mutations affecting sensory receptor proteins that tune perception to low-light conditions. Comparative genomic studies across species with different activity patterns have begun to identify the genetic changes associated with evolutionary transitions between nocturnal and diurnal lifestyles, revealing patterns of gene loss, duplication, and modification that track with shifts in temporal ecology. The genomic signatures of the ancestral mammalian nocturnal period remain visible in the genomes of living mammals, including gene losses affecting visual pigments that were apparently dispensable during the nocturnal bottleneck, but could not be recovered when lineages subsequently returned to diurnal activity. The application of environmental DNA methods to the study of nocturnal animals represents a recent development that may prove valuable for detecting rare or elusive species whose presence might otherwise go unrecognized. Animals continuously shed DNA into their environment through feces, urine, skin cells, and other biological materials, and the collection and analysis of environmental samples can reveal which species occupy particular habitats without requiring visual observation or capture of the animals themselves. For nocturnal species that may be present but undetectable through conventional surveys, eDNA methods offer a complementary approach that may prove especially useful in cryptic species or those occurring at low densities. The scientific understanding of nocturnal animals has developed within broader intellectual contexts that have shaped the questions researchers ask and the frameworks within which they interpret their findings. Early natural historians documented the habits of nocturnal creatures with varying degrees of accuracy, and often embedded their observations within cultural narratives that associated darkness with danger, evil, or the supernatural. The gradual transition from natural history to scientific zoology brought more systematic approaches to documenting nocturnal animals, though the practical difficulties of studying species active at night continued to limit knowledge relative to diurnal species. The development of evolutionary theory provided a framework for understanding why different lineages had adopted different temporal niches, and the ecological concept of the niche provided tools for analysing how species partition resources, including time. The investigation of sensory physiology in nocturnal animals contributed importantly to fundamental understanding of sensory mechanisms that extends beyond nocturnal specialists to illuminate general principles of sensory function. Research on owl auditory localization, for example, revealed principles of neural computation for sound localization that have implications for understanding auditory processing across vertebrates, and studies of bat echolocation have contributed to understanding of acoustic signal processing. motor control, and neural plasticity. The choice of nocturnal species as model organisms for particular research questions has thus had consequences for fields extending well beyond the ecology of nocturnality, with findings from these species informing general biological theory. The temporal organisation of ecological communities involves far more than simple separation between diurnal and nocturnal species, with fine-grained temporal patterns structuring interactions within both day-active and night-active assemblages. The activity peaks of different nocturnal species may be offset from one another, reducing competition for resources or predation risk through temporal niche partitioning at scales of hours rather than the entire night. Seasonal changes in night length alter the activity budgets of nocturnal animals, potentially constraining foraging time during short summer nights in high latitudes, while providing expanded activity periods during long winter nights that may coincide with reduced food availability and increased thermoregulatory demands. The interplay between photoperiod, food availability, temperature, reproductive demands, and predation risk creates complex constraints on the temporal organisation of behaviour that researchers have increasingly sought to understand through integrated approaches, combining field observation with physiological measurement and mathematical modelling. Climate change poses potential threats to nocturnal animals through multiple pathways, including direct physiological effects of altered temperature regimes, indirect effects mediated through changes in food availability and habitat structure, and shifts in the timing of seasonal events that may disrupt previously synchronized relationships. The photoperiodic signals that cue seasonal behaviors in many nocturnal species operate independently of temperature, creating the potential for mismatches between photoperiodic cues and the now shifting thermal and phenological environment. How nocturnal species will respond to ongoing climate change remains uncertain, with predictions depending on factors including the flexibility of behavioural timing, the breadth of thermal tolerance, and the potential for evolutionary adaptation over timescales relevant to current rates of environmental change. The urban environment presents a novel selective landscape for nocturnal animals, with the combination of abundant resources, altered predation pressure, light and noise pollution, and modified habitat structure creating conditions that favour some nocturnal species while excluding others. Urban adapted nocturnal species, including various rodents, raccoons, opossums, and certain bats, can achieve high population densities in cities, exploiting food resources derived from human activities and finding shelter in the modified structures of the built environment. The behavioural flexibility that allows some species to thrive in urban environments, while close relatives remain restricted to less modified habitats, represents a subject of growing research interest, with studies examining whether urban populations show genetic differentiation from rural populations and whether urban environments are driving evolutionary change in the species that have successfully colonised them. The interactions between nocturnal animals and human activities encompass both conflict and benefit, with nocturnal species variously regarded as pests, threats, resources, or valued components of natural heritage depending on context and perspective. The damage caused by nocturnal rodents to stored food and agricultural crops motivates extensive control efforts worldwide, while the predation on livestock by nocturnal carnivores generates conflict with pastoral communities in regions where large carnivores persist. Conversely, the consumption of pest insects by bats and the pollination and seed dispersal services provided by various nocturnal species represent ecosystem services of substantial value that may be inadequately appreciated or incorporated into land use decisions. The hunting and trapping of nocturnal animals for food, fur, traditional medicine, and other purposes has historical significance in many cultures and continues to affect populations of target species, with sustainability depending on harvest levels relative to reproductive capacity and the protection of critical habitat and refuge areas. The cultural representations of nocturnal animals have varied enormously across societies and historical periods, with nocturnal species figuring prominently in folklore, mythology, and symbolism in ways that both reflect and shape human attitudes toward these creatures. Owls have been associated with wisdom in some cultural traditions, and with death or ill omen in others, while bats have been viewed as symbols of good fortune in Chinese tradition, but associated with vampirism and supernatural evil in European and American popular culture. These cultural associations can influence conservation attitudes and the willingness of communities to tolerate nocturnal species in their environs, creating both opportunities and challenges for conservation efforts that must operate within cultural as well as ecological contexts. The representation of nocturnal animals in scientific discourse has evolved alongside changing methodological capabilities and theoretical frameworks, with contemporary research emphasizing the integration of multiple approaches and the consideration of nocturnal species as members of complex ecological communities rather than isolated curiosities. The distinction between nocturnal and diurnal species, while useful as a first approximation, proves increasingly inadequate as research reveals the complexity and flexibility of animal temporal organization, and current work increasingly focuses on the mechanisms and consequences of temporal niche differentiation at finer scales of resolution. The continued development of technologies for studying animals in darkness, combined with the integration of physiological, behavioral, and ecological approaches within evolutionary frameworks, promises further advances in understanding of how and why animals have organized their lives around the cycle of light and dark that has characterized Earth for billions of years. The investigation of nocturnality in animals connects to fundamental questions about the organization of life on a rotating planet with its inherent cycle of illumination and darkness, a cycle that has shaped the evolution of virtually all organisms from the earliest stages of life's history on Earth. The circadian rhythms that govern temporal organization in bacteria, plants, fungi, and animals all represent responses to this fundamental environmental periodicity, and the understanding of how organisms have variously accommodated to and exploited the light-dark cycle provides insight into the relationship between environmental constraints and biological diversity. The nocturnal animals that occupy the night represent not a uniform category, but a diverse array of lineages that have independently evolved solutions to the challenges and opportunities of life in darkness, and the comparative study of these solutions reveals both the contingency of evolutionary history and the consistent patterns that emerge when different organisms face similar selective pressures. The ongoing investigation of nocturnal animals continues to yield discoveries that challenge previous assumptions and reveal unexpected complexity in the sensory abilities, behavioral repertoires, and ecological relationships of species that have long remained hidden from human observation by their preference for activity in darkness. The technological advances that have progressively expanded the ability to observe and study nocturnal animals promise to continue accelerating the accumulation of knowledge, while theoretical developments in ecology, evolution, and physiology provide frameworks for integrating this growing body of information into coherent understanding. The nocturnal realm that encompasses roughly half of each day across the surface of the Earth sustains a diversity of animal life that rivals and in some respects exceeds that of the diurnal realm, and the scientific exploration of this nocturnal world remains an active frontier of biological research, with much still to be learned about the remarkable animals that call the night their own.